That's fine. I usually stay after Rick Evans anyways.
All right, continuing on with chapter 33. Hopefully we finish it today. Hopefully. Okay, so we've been discussing animal diversity, and this next section was about animal phylogeny, putting together the tree. I think we took notes on this slide already about how there's little disagreement about where we place animals in the phylogeny or the tree of life. Traditionally, they looked at features of anatomy as well as embryological development. But now with genome sequencing, it has changed a little bit, and I'll talk about some of those changes. But this is essentially kind of the timeline that we're going to work our way through. And so Metazoa means animals, and we'll start discussing sponges, and then we'll move on to our radial organisms, to no body cavity, to somewhat of a body cavity, to finally a full body cavity, and then some more unique features of animals. Okay. So the current understanding does differ from traditional views. Like I said, the phylogeny or the tree of life, those cladograms, they're basically a hypothesis. And whenever we get new data, we challenge our current views and make adjustments as we see fit. Some consistent results, okay, Porophora, sponges, are a monophyletic group. So based on their characteristics, we thought they were monophyletic, but now with genome sequencing, we've confirmed, yep, they're a monophyletic group. Cnidarians, okay, jellyfish, they branch off from the tree before the bilateral organisms, bilatera, and that's consistent. Deuterotome branches within our bilatera branch, if you will. However, there have been some inconsistent results with the protostomes a little bit. So you'll see some shuffling around with that. Annelids versus arthropods. Annelids and arthropods both have segmentation, and so we actually grouped them together, or like right next to each other in the tree of life. But now they do belong to separate clades. Arthropods are grouped with protostomes that molt their cuticles at least once during their lifetime, so they're actually a part of the Ecdysozoans, and annelids do not do that. In one of our radial organisms, the Ctenophoria, so we'll talk about the cnidarians and then the ctenophores. Ctenophores can be both diploblastic or triploblastic. There's like inconsistencies as to whether or not they're radial or bilateral. So when I show you the tree of life, you can just kind of see that. They don't really know. They just think it's in between cnidarians and the bilatera for sure. And then the Chaetonathia is considered both a coelomate, something that has a full body cavity, as well as a pseudocoelomate. Don't ask me how that happens. So there are still some inconsistent results due to genome sequencing, and hopefully we can, finalize or streamline or come to an agreement in the near future.
How can you be both triploblastic?
I think, you know what, honestly, I don't even know. I read this in the book. This is something that's new in this edition, and I'm like, what? I think it has to do with their symmetry, because they're not technically radial symmetry. If you have radial symmetry, then you are diploblastic. But they also are not bilateral. They're kind of like they're neither. I don't know. So to me, they look bilateral. So then they're like, okay, we should call them triploblastic, but they don't really match up with all the other features of a triploblastic organism. So they're just, I don't know. Messy, messy, messy. All right. Bless you. So the traditional view and what the old edition covered the organisms, we started with acoelomates and pseudocoelomates, and it was literally a chapter in the book called non-coelomates. So they combined the two in one chapter. And then they had a chapter on the coelomates, which was obviously very long and extensive. So that was the traditional view. So now we're not going to do that. We're going to start off with radial, then jump into bilateral, and then go into acoelomates to pseudo to coelomates. So a body cavity, a coelom, does appear to have evolved just once. We do find some clades that have reverted back to a no body cavity, and you'll see that noted in the tree, the animal tree. You also see pseudocoelomates nestled in with the coelomates as well, so it kind of goes both ways with both acoelomates and pseudocoelomates. But Deuterotomes, all of them have coeloms, for sure. All right, we're going to move on to protostomes here. So protostomes, remember, what does the blastopore become in a protostome?
The mouth.
The mouth, okay? And so we can categorize protostomes as either spirillans or spiralians, I think I'm saying that right, and ecdysozoans. So spirillans means that they have the spiral cleavage, and that could be further subdivided into two more groups, if you will. Lophotrochozoas and the Plateozoa. Lots of big words in this chapter, Lophotrochozoa. Some basic characteristics of Lophotrochozoas, they move by muscular contractions. They also have something called a lophophore, and so we'll talk more about these in depth, I think, in the next section, or it's coming up here soon. Plateozoas don't have a body cavity, and they move by ciliary action. Second group underneath protostomes is the ecdysozoans, and these are organisms that molt. We'll go through ecdysis. So you can see this. I don't know what kind of insect that is. I don't think that's a cicada, no. But they molt. They'll shed their exoskeleton. Arthropods are the largest for the ecdysozoans. So, yes. So protostomes divided into two clades, spirillans and ecdysozoans. Spirillans subdivided into Lophotrochozoas and the Plateozoas. Oh, of course. All right, can I move on for a quick note about deuterostomes? All right, so deuterostomes are going to include the chordates, chordata, and the echinoderms, spiny skin. So most of the animals are protostomes. Deuterostomes have fewer phyla and species. But the deuterostomes, believe it or not, are more uniform in many ways, despite a lot of the differences in appearances. And so we'll cover that, I think, in chapter 35. So kind of examples of echinoderms, just throwing it out there, like starfish. Yeah, all I can think of is starfish at the moment. Sand dollars. So this was the pathway that we covered before. This is animal, okay, monophyletic. We believe that it evolved from some type of colonial flagellate that's very similar to the flagellates of the protist, because you'll see similar structures in their sponges, porophora. And then we went on to the true animals that have true tissues, to Cnidaria, to Notophoria, and then jumped into deuterostomes and then protostomes. But here's the new way that we're going to cover it. So we're going to talk about sponges. I'm not going to get very specific about species of sponges, but just know that there are actually two kind of clades within that. But then we're going to jump into Notophoria and Cnidaria. That's the same, but then we'll jump into bilateral, and we'll cover the protostomes first, and then we'll come back and cover the deuterostomes. So you can see that, see the differences just a little bit. So we subdivide our protostomes into our Lophotrochozoas and our Ichthysians. Here is just some of the phylums that we are going to cover. For sure, arthropods, mollusca, chordata, platyhelminthes, nematoda, annelids, cnidarians, actually all of these. There's the first two up here. Brachiopodia we'll cover. I'm not sure about this one. Ctenotophoria, I'm not sure about this one. For the rest, none of these down here. But we're going to cover a lot of these, and this is just listing 20 of the 35 to 40 known phylums of animals. So lots of animals to cover here. Review questions. Which of these is not a protostome? D, Echinodermatata. Okay, so that's obviously a echinoderm. What has recent molecular data revealed about the phylum Porophora? Something that's consistent.
Is it B?
Nope.
A?
It's A. They're monophyletic. Sponges are monophyletic. Recent molecular data has led to a revision of the traditional classification of animal phyla. The annelids and arthropods are now moved into different clades, and the arthropods are now assigned to blank animals that molt. B. Ichthysozoas. True or false? It is accepted that the animal kingdom is monophyletic. True. What do deuterostomes not include? B. Ichthysozoans, because those are protostomes. Very good. What clades do Spirulia comprise? E. Lophotrochozoas and Plateozoas. Nicely done. All right, so let's talk about sponges, parazoas, animals that lack specialized tissues. So I'm going to talk about some of their specialized tissues, explain a little bit about their cyanocytes, these cells that look like the cyanoflagellates, the protists that we discussed a few chapters back. So porophora, or sponges, do have a loose body organization. But first off, let's talk about their symmetry. They have none. Okay, so there are a few that are radially symmetrical. Most of them don't have any symmetry. They're just random. Believe it or not, sponges in their early stages, their larval stages, are free swimming. So they'll drift in the water, they'll swim through the water, and then when they are anchored or become anchored, then they'll develop into their adult form where they can't move anymore. So they are sessile. This is new information that I did not know. There are some species of sponges that actually produce chemicals to defend themselves, to repel against predators, as well as organisms that might try to overgrow or take over their area. And so there's been a lot of research with sponges about these chemicals. Maybe there is some type of pharmaceutical value to them. So I was like, well, I didn't know that about sponges. Yeah, so I guess I should say just from now on, from the rest of this lecture on to chapters 34 and 35, this is where we're starting with the animal tree. We're starting with the phylogeny here, okay? So we're going to tackle the parazoas. This is obviously just the sponges. And then we will move on to the eumetazoas with the cnidarians. Okay, so let's take a look at the special cells that some of these, or I should say sponges, have. Okay, there's a lot of different cell types. You know, despite being a sponge, you know, they don't have any heads, no appendages, no mouth, anus, or any type of internal organized structure to them like all the other animals have. But they do have some cell types that carry out special jobs, like cells that rapidly contract the oscula, which is the opening pore on the top. Cells that help synchronize reproduction. So there's some type of cell communication going on. They have cells that make up their structure. We call them spicules or spongin, so they can build complex organized meshworks. So I mean, it seems to be more than just a mass of cells embedded into some type of gelatinous matrix. It's way more than that. Remember with sponges, does anyone remember the reason why sponges don't have like true tissues? Yeah, they can go back and forth. We call that dedifferentiate. So they can dedifferentiate from a specialized state to an unspecialized one. And so there's been experiments where they've taken a sponge and they've put it through like a coarse cloth or sieve, kind of like a microscopic like filter, to separate all the cells. And those cells will actually actively seek each other out and reassemble the sponge. Kind of cruel to do, but yeah. All right, so we're going to jump into, in detail here, the specialized cells, the cyanocytes, the spicules, and the spongins. So when you take a look at the walls of a sponge, you can see it's got 3 layers. So we're going to start, I guess the first one is the inside layer, made up of cyanocytes. So they face the internal cavity. So this is an open cavity. There's an opening at the top called the osculum. And sponges have pores in them. I mean, obviously sponges, just think about a sponge. Some of, the sponges that you buy in a store, sometimes they were living organisms. But what happens is that water filters through these pores, okay, and will converge at the osculum and be expelled out. And so these cyanoflagellates, they flap their little flagella, and the food travels through, and it can get caught in the mucus, and then we have phagocytosis that occurs, and that's how sponges eat. So there's a limitation here with cyanocytes. They can only eat like microscopic organisms, bacteria, small food particles. So that whatever they eat is obviously smaller than this cyanocyte. On the outside, we have an outer epithelial layer of these flattened cells, as you can see in orange here. They help comprise the pores. Your book also calls the pores ostia. And then the middle layer, the mesohyl, is rich in protein. It's like a matrix of other cells, but it's 2 types of materials. I guess I don't even know if I should call them cells, but spicules and spongins. So spicules are made up of calcium carbonate or silica. Remember, there's like 2 different clades within sponges, those that produce calcium carbonate and those that produce silica. So these spicules are either calcium carbonate or silica. And then we have spongin that kind of fills in between, made of tough protein. So the spicules here are in yellow. And do they show the spongin? I'm assuming. No, that's not the spongin. Spicules. Anyone seeing spongin?
No.
No? Okay, so they don't show spongin on this one. I guess I should talk a little bit about this cyanocyte. So it does resemble that protist, okay? Beats its flagella, helps drives the current of the water, if you will. These little guys, yeah, for being how small they are, they can actually move a lot of water. I think your book talks about a statistic, 1 centimeter cubed. Yeah, because it's 3D. So 1 centimeter cubed of cyanocytes can filter 20 liters of water a day. So I think that's crazy for how small these guys are. So, okay. So sponges can reproduce asexually as well as sexually. How they do it asexually, if sponge breaks off into fragments or pieces, then they can become individual organisms. Sexually, we have male and female parts here. So, I have a typo. Sponge sperm created by. It's supposed to be created by, not created. Sponge sperm created by the transformation of a cyanocyte. So a cyanocyte that de-differentiates into sperm is released into the water, and that sperm will be captured by another cyanocyte and carried to the egg cell, which is held in the mesohyl. I really should look up how to say that word. In some species, that development will occur within the mother. In other species, it might release the fertilized egg, and then the sponge sperm might find that egg in the water and fertilize it there. So a couple different ways that sponges can reproduce sexually, but just know that they can do both. And that does it for the parazoas. Okay, before animals. Sponges. Okay, moving on to some review questions. How do sponges differ from all other animals? What? B? Okay, I guess that's kind of true. I was looking more for E, but you're right. I think I couldn't, yeah. Terrible question, my bad. Moving on to, so just to clarify, yes, they don't really have specialized tissues. I mean, they have specialized cells, and so, yeah, they don't have tissues because they can de-differentiate. I was actually looking for E, but you're right, they don't have tissues. So the reason why they don't have tissues is because they can de-differentiate, right? Okay. Which of the following is characteristic of sponges? B, sexual reproduction, okay? Adult mobility, no. Their adult forms don't move, but the larval stages do. They don't have tissues, organs, no symmetry, so I guess B is your best option. Using that ACT science prep. Select a statement that is not true concerning cyanocytes. Crickets. It's C, yeah. So consume particles many times their size. That means larger than themselves by stretching their cells. So no, that's false. They can only consume particles that are smaller than the cyanocytes. Spicules and spongin are found blank of a sponge. B within the mesohyl. I don't know. I should probably.
That's such a weird word.
Yeah, I feel like that would be a great hangman word, you know, and you just really mess people up on that. Kind of like the word lynx. That's a good one too.
Links?
Lynx. There's no vowel.
Oh.
Yeah. All right, Eumetazoas, animals with true tissues. So we're going to start off with cnidarians and then transition into ctenophores and then discuss the question of symmetry with those ctenophores. Are they bilateral organisms or radial organisms? I don't know. So the first key transition, remember, is distinct tissues. That's why we call them Eumetazoas. We got to go back to embryonic cell layers, okay? How they differentiate into tissues of the animal body. Remember with the germ layers, there's 3 of them, the ectoderm, the mesoderm, and the endoderm. Well, we're going to discuss organisms that just kind of have 2, basically radial organisms, ectoderm and endoderm. They lack a mesoderm. If you have a mesoderm, then you are bilateral most likely. So our ectoderm is the outer covering, basically makes up the epidermis as well as the nervous tissue. And then the endoderm is going to form like digestive tissues, kind of like the inside, or sometimes we call digestive tissues gastrodermis. And if you have a mesoderm, then it will be between the ectoderm and the endoderm and help form muscles or the skeletal system. So today there are 2 radially symmetrical, and see now I'm calling them radial here, but that question of the ctenophoria, the comb jellies that exist today. So I'm going to start off with the cnidarians. We know for sure are radial. Some examples of cnidarians: hydroids, jellyfish, sea anemones, corals. And then I'm going to talk about a unique organism called the ctenophoria, which is our comb jellies. Now, the thing I want to emphasize with these radial organisms, they have distinct tissues, as I have listed on this slide, as the first key transition, but they don't have organs per se. Okay, so we'll talk about if they don't have organs, well then, how did they digest? How do they carry out some simple functions? So we'll talk about that. Now you'll notice that I have color-coded the cnidarians in blue here and the ctenophoria as purple. You're going to see that for the rest of this unit. So colored slides are important. Now colored slides just mean that it belongs to that phylum. So if you see a slide in blue, that means it's cnidarians. And if you see it in purple, then we're talking about ctenophores, just because we're going to be discussing so many different animals. It's just going to be all over the place. Thank you. All right, can I, nope, it's fine. So I don't know what you want to do with your notes, if you just want to keep them in black or somehow denote that this is about cnidarians and then this is about ctenophores. I don't know.
So we thought that it's not that severe.
All right, so the basic body plan of cnidarians, our first blue slide here. So they're kind of gelatinous in their composition, okay? They are carnivores. They eat their prey using their tentacles that ring the mouth. They do have 2 different body plans. They have something called the polyps. can see that in this diagram down here. And the Medusa stage, which is what you're probably more familiar with. So despite having no reproductive system, no circulatory system, no digestive system, no excretory system, cnidarians can reproduce, they can exchange gas, they can actively capture and eat their prey, and distribute, you know, the food that they get from their prey to all of their cells. So it's kind of amazing, actually. The two basic body plans, the polyps and the medusa, they are quite different body forms, but they do share a lot of the same, as you can see here, morphology. Gastrodermis, epidermis. You got a mouth, there's the mouth. So that's kind of cool. You'll notice that the mouth opens up into the gastrovascular cavity, this kind of dark shaded purple. And over here, dark shaded purple, mouth opens up into the gastrovascular cavity. Now at the polyp stage, they are attached to a substrate. So substrate's like a rock or some type of structure. The opening acts as both mouth and anus. It's faced upward. Medusa is the free-living stage. You know, kind of goes with the ocean currents, can pulsate using water propulsion. Umbrella-shaped, tentacles surrounding that mouth. If you take a look at the family tree, we just discussed the Paraphora, the Parazoas, and so now we are talking about cnidarians. Cnidarians are not protostomes or deuterotomes, okay, so they're like before that. So we just call them animals with true tissues, the Eumetazoas. Okay, this next slide, another picture showing the difference between polyp and a medusa stage. So here we have a sea anemone and its polyp stage. Again, gastrovascular cavity opening to the mouth and anus. Here's a jelly, the medusa stage. So gastrovascular cavity is somewhere right here. So let's talk a little bit about that gastrovascular space. This is where digestion occurs, where it exchanges gas, waste discharge, formation of gametes. So a lot happens in that gastrovascular space. It can also serve as the hydrostatic skeleton. And the purpose, two purposes of that hydrostatic skeleton. Obviously, it's going to give it a little bit of a structure so muscles can operate, and then obviously give that animal its shape. Okay, so the gastrovascular space, just out like what makes up the boundary of that, we have the gastrodermis, and then the mesoglia, and then the epidermis on the outside. So if I go back to this picture, you can see there's the mesoglia in yellow. Nope, it's this light blue area. The gastrodermis is in yellow, and then this dark blue is the epidermis. So the mesoglia is space between those two layers. Life cycle, it varies depending on the species. You know, some, they only occur as polyps, kind of like the sea anemone. Some, it's straight up medusa stage. Some actually flip-flop between the two or alternate the two phases, but I just want to emphasize that both of these polyps or the medusa stage or phases are diploid. So we don't have that alternation of generation going on because you went from haploid, multicellular haploid to a multicellular diploid. These are both diploid phases that are obviously multicellular. The polyps, if you're in that polyp stage, they can reproduce sexually or asexually. So with the asexual, it might just like bud off or just break off and form a new polyp. But then they say, hey, sometimes these polyps can actually produce a medusa stage. And I'm like, oh, okay, so that's, even more confusing. So am I really going to quiz you on the life cycle of cnidarians? No, just because they're so vague. They're just kind of saying, hey, there's some species that do this and some species that do that. But I will, for some reason, your book thinks it's really important to know that the larval stage of a cnidarian, before it becomes a polyp, is called a planulae. So if anything on this slide, this bullet point right here, number 3, is the most important bit of information. If it's in the medusa stage, then they can only reproduce sexually. Or at least that's what I interpreted from the book. So. Okay, so moving on to how does the gastrovascular space, carry out all of these very important functions for the cnidarians. Let's talk about digestion here. So one of the key evolutionary innovations for animals is that we start to see some internal extracellular digestion of food. Whereas with the sponges, they kind of just like filter feeded and used phagocytosis. But here now we actually have some like extracellular digestion of prey. So it takes place within that gastrovascular cavity. And once it consumes its prey, brings it in using maybe tentacles, and it passes it through the mouth, we have these cells that line the gastrodermis that actually release digestive enzymes to break down the prey, to break down the food. And then again, using phagocytosis, they will bring those food particles in to the cell. And so with sponges, they could only eat prey that was smaller than them, but now we have the ability to eat animals that are larger than the individual cell. Okay, one more unique feature about cnidarians. Why are they called cnidarians, basically? So cnidarians actually have a specialized cell called Nitocites. Okay? Nitocites, specialized cells. And one type of specialized nitocyte is called a nematocyst. So if I'm looking at this polyp here, and we zoom in on this tentacle, you can see we have some cells that are different from these other cells. And these, I don't know, V-shaped cells is actually an example of a nematocyst. Nematocysts, on the inside, they have this harpoon-like structure that shoots out, grabs hold of their prey, coats it or injects it with toxins to subdue their prey, and then their prey is, paralyzed or doesn't move, and then they just reel it on into the mouth to be ingested. Now, because the mouth serves as the anus as well, they can't eat and digest at the same time. They got to bring that prey in, then they'll digest everything, and then they excrete it out. Whereas us, you could eat and digest and eat, and you can do both at the same time here, because everything is in one spot and they only have one opening for the entry and exit. They gotta make time for it. So capture their prey, bring it in, digest it, get all the nutrients, and then get rid of it. But nitocytes or nematocysts, these are specialized cells. This is why jellyfish sting, because they got stung by nematocysts. Okay. I would say by far nematocysts, probably one of the most complex structures secreted by single animal cells. So it's a nice little setup there. So what are some species that have these nematocysts? Well, there's 5 classes. The last edition only had 4 classes, and now we have a 5th class. So it's amazing what can occur in the last 15 years. So the first class that we'll discuss is called Anthozoas, sea anemones and corals. They are plant-like. Antho kind of means flowery. So they're plant-like, or you look at them and they just kind of look like plants on the ocean floor. Their polyps tend to differ from the polyps of the other classes. Some other differences: we see that their gastrovascular cavity has mesentery, which is a clear tissue on the inside, so we see some compartmentalization going on here. Their tentacles are hollow, whereas with other cnidarians, they're solid, filled with Mesoglia or other tissues. Coral reefs fit underneath the class Anthozoas, and they're economically important. They serve as homes for crustaceans and fishes, lots of different organisms. Not only that, coral reefs, they protect coastlines. They kind of reduce the damage of tropical storms, kind of take the brunt of it. So they protect the coasts of tropical islands. However, they're not doing so great right now. So there are some parts of the ocean that are becoming warmer. So we talk about global climate changes. Warmer water, what's the big deal about warmer? Well, first off, coral reefs are found in warm water. But when it reaches past a certain threshold, then the symbiosis between a zooanthozoa and an algae, is it an algae? I think it's an algae. It starts to break down, and we start to see coral bleaching. I'll just finish this slide here. So as CO2 levels rise, CO2 increases the acidity of it, so the water becomes more acidic, and acid breaks down calcium carbonate, which makes it less available to build the corals. Okay, so I'm gonna exit and stop here, and we'll just.
Wait, can I take a picture of this?
I'll show this slide again. I'll start it again. I'll recap. So.